This is a production of Cornell University.
Thank you very much, Mike. And thank you very much for the invitation. Thank you, Ed and Liz, for being fantastic hosts. And I'm very happy to be here presenting this talk in Cornell with a beautiful fall day. So my talk is going to be about improving plant breeding efficiency with quantitative genetics. So I need to talk to this side, right? Okay, so we'll see how we do. That's better, right? Okay. So what I'm going to be doing is I'm going to go through a few projects that we have been working on and how we're using some of this quantitative genetic theory, some of these tools to actually ask questions that are relevant for breeding programs and for improving efficiency in breeding programs. So I do like to start with this response to selection equation. I think this is not going to, I'm going to move it. So is that better? Yeah, I think so. Okay, so with a response to selection, we start with a population that has a mean, and we select individuals, and hopefully we would like that the next generation moves forward in the direction of where we selected. And how much do we move that population depends on a few things, and that is mainly the intensity of selection. So how many of those individuals do we select? The variance of that population, in this case, the genetic variance that we put there, and then, or the additive variance, and then the accuracy of how good are we at identifying those best, genetically best individuals, right? And that accuracy could have to do with, if we're doing phenotypic selection, has to do with the heritability, but there are other ways we can evaluate that accuracy. But bottom line, it's a function of this population, of how much variation do we have in this population, how much pressure do we put into the selection, and how good are we at selecting? And I'm going to show you how we can impact this at different levels when we are working with our optimization strategies. We're going to be using some strategies that are going to improve the intensity of selection because we can have larger population sizes, or that is going to improve the accuracy of the selection. So we can work on different aspects for that. But before we go there, we need, we're going to be evaluating these individuals based on their phenotype. So what is this phenotype? If we have two different individuals, we have the genetic component that is relevant, and that is going to affect the phenotype. But we also have the environment that will affect this response and how we perceive the phenotype. And then we have the interaction between this genotype and the environment. And all of this is gonna provide us the phenotype. So how do we evaluate and how do we control or measure or utilize all of these components is very important in being able to identify the superior individuals. So this is the first basic equation of quantitative genetics, that is the phenotype is a function of the genotype, the environment, and the genotype by environment interaction. So this environmental variation, right, that causes the same genotype to have different performances in different environments, we can separate it in two components. One is what we call the microenvironment. And this is the environment associated to a single plant or a single plot. And we know that, the same genotype, if we can replicate that genotype planted here and there, it's going to provide different yield, or it's going to have different plant height. So the phenotype is going to be different from plant to plant or plot to plot, right? And that is what we call the microenvironment variation. And we can control that with proper experimental designs. You're probably all very used to using different experimental designs for controlling that microenvironment. But we also have the macro environment, and this is the variation or the environment associated with a location and time. So these are my location and year combinations for the evaluation of the same genotype, right? We're assuming we can replicate this genotype and we can evaluate them in multiple environments. Why is that relevant? Because from year to year, we're going to have a genotype by environment interaction. So that means that the top ranking genotypes in one environment might not be the top ranking genotypes in another environment. And that is very relevant when we do selection, okay? So if there was no G by E, then it's just an environmental effect and we can select in any environment and that will be fine. But if we have this crossover interaction, then we need to be careful in how we model, how do we deal with this G by E. So the first part is I'm going to be talking about how to control this microenvironmental variation. And this is a work by one PhD student, Alejandra Borges, in which what we were interested was to compare these experimental designs for large genomic studies. And there has been some studies in the 50s using some uniformity trials where they were looking at this experimental design optimization, but they were not addressing the newer experimental designs that we can use now that we have hundreds of genotypes to evaluate in the same trial. So we wanted to see how we could improve on that. So what we took is a very large field of wheat. So these were 64 hectares or 158 acres that were harvested in rectangular plots. And then on top of this, what we did is we simulated different experimental designs. So we simulated some of the most commonly used experimental designs, and those are the completely randomized design here, the randomized complete block design here, the alpha, which is an incomplete block or alpha designs with different number of experimental units within a sub block. And then we also use this P-rep, and these are augmented designs. So these are not in this augmented designs, not all the genotypes are replicated. Some of them are replicated and some are unreplicated. So they are called the P-rep designs. In this case, the G uses the same number of genotypes as all these other designs. So let's say we were working on the 50 experimental design, 50 genotypes. This had 50 in 3 replications, 50 in 3 replications, 50 in 3, all of those. This one also had 50 genotypes evaluated, but because some of them are not replicated, the number of experimental units is smaller, okay? Now, the other thing is we also evaluated this P-rep N, assuming that, okay, what if we keep the number of experimental units constant, and this is not a function of we are just interested in evaluating this population that has 50 genotypes, I can pull individuals from this larger population that will make sense to evaluate, and I'm limited by my resources in evaluation, and that it means how many experimental units we can have. And that is the P-rep N. So this one has more genotypes evaluated than those 50. So it's trying to make comparisons a little bit fair for the different experimental designs. We did this for 50 genotypes. We did it for 200 genotypes, and we did it for 15. And that was to have a sense of, because, you know, the efficiency of the experimental design is going to depend on the size, the actual size of the experiment. In smaller experiments, it's easier to control for the spatial variability. So that was the logic. And here is the result for the medium size for the 50 genotypes. And what we have here is, I'm showing you the correlation. This is the correlation of the true genotypic value with the predicted by the model, the genotypic value predicted by the model. And then we have for each one of the experimental designs, we have a no spatial correction. So besides randomizing the experiments in different ways, we also compare no spatial correction a posterior. Then we compare with an AR1 model. That means that we're modeling a one-dimensional process in the columns. And then this is an exponential, so it's a two-dimensional model in which we are modeling the correlation among experimental units. And this comes because we know that two plots that are close together tends to be more similar than two plots that are further apart. So we were doing this post-blocking or post-spatial corrections. And this is shown for each one of the experimental designs. And you can easily see that the alpha experimental design was the best one, okay? And this is, okay, you know, not so fireworks, not so many fireworks, but people have been reporting that alpha designs are better, but they haven't been compared to this P-rep designs, and this is for a single location, right? So for me, this was very important because there are tons of genomic studies that are using these augmented designs, and you can see that there is a compromise there on the correlation that you get of the true predicted values and the on the true values and the predicted values from the model. So there is a compromise that you have here. Yes, on this one you can use fewer resources. On this one you're still using the same number of resources, but you get a compromise here. The advantage of this one is that maybe you're testing more individuals. So we're gonna finish the story with another paper, but for now, just keep in mind that the alpha design was the best one, and that was for high variability areas within that 64 hectares, and also for low variability areas. And we sampled 100 independent sites within those locations so that we will be covering the whole area. So key messages from here is that the best experimental design was the alpha. The second key message is, if you look, there is always an advantage of modeling the spatial correlation. So there is always an advantage of using that pause blocking. The thing is that with the spatial correlation in this, there is no specific design, you do not get as good as with an experimental design. So spatial corrections are good, but cannot be used as a substitute for my experimental design. And that was something that was very important, because there is all this, theory that now with all these tools that we have for modeling the spatial variability, why on earth would we do experimental designs? And this is the reason why we would do them. So this is just sitting down before you do your experiment and thinking about it for a few minutes. It doesn't take money, it doesn't take any extra cost. It's just thinking about it beforehand. And there you go, you get a huge improvement in the prediction, in this case, in the prediction accuracy. You also get a huge improvement in the recovery of the top 15% of the genotypes. We were also looking at model fit, and we were also looking at standard error. So we're better able to compare or to detect differences among genotypes when we have this type of experimental design. So this was a nice story for that microenvironmental variation. And then we continue to this macro environment, and then I'm going to show you how we merge those two. So this G/E, if we have one environment here, genotypes performing here, genotype 1 is better than 2. In another environment, genotype 2 is superior. This is a classic way of crossover interaction where the winning genotype is different depending on the environment. And this is mainstream. This is very common in plants. We have it, we see it all the time. So how do we deal with this? There are a few, and I'm going to show you just a few areas where we have been doing this modeling so that we can actually exploit the G/E instead of assuming that it's not there, or that it's very little or marginal. So the first one was to compare strategies for exploiting this G/E when we map, when we're doing QTL/E. And I'm talking about QTL/E in a broad sense, not necessarily as a biparental QTL mapping, but GWAS, just mapping quantitative traits. So if we have the phenotype will be a function of the overall mean, the environment, the genotype, and the G/E. We can now dissect this genotype into two components, and one is the marker effect, and the other one, so we will have a marker effect and all the other genotypic effect that is not accounted by this marker. And then the G/E will have a marker, an environment-specific marker effect, and all the remaining G/E that is not accounted by that marker. If we combine this, assuming that we have a table of G/E means and we do not have the raw data, then we can have a single test for whether there is a main effect QTL or an environment-specific QTL. And that will give us a peak on either one of those situations, and then we can separate whether it's a main effect or G/E, but we can do the genome scan with this very simple model. So now we have this model for modeling the QTL/E. And I'm going to show you one of the results of using that model here. This was a mapping population, a GWAS population in barley, where we were looking at 450 genotypes evaluated in multiple environments. Here are environments, and here are QTNs. And this was for leaf rust. And you can see that there is this QTN that was, global. So it had an effect, but the effect was consistent across the environment. So this is kind of like the breeder's dream, right? We can deploy it in breeding because it's an allele that is going to be good in all the environments. Then we had this one, that there was G by E, but only magnitude differences because everything is blue. So that means that the good allele was good in all the environments. Again, you know, breeder stream, we can work on this. And then we had this other QTNs that had a G by E of crossover interaction or marker by environment crossover interaction, where in some environments, one allele was positive and the same allele was negative in the other environment. So in these situations, it is not the breeder stream in the sense that we cannot readily deploy it, but it's the physiologist's dream, right? That what is going on that is causing this. And so we have been working on this. This has to do with the mega environments or groups of environments. So this is for the phenotype per se, not for the marker. It's for leaf rust. So groups of environments that have low G by E for leaf rust and the other group of environment. So this has to do with, you know, race specific, with temperature, with environmental conditions that cause this pathogen to act different or to have a different performance. So this is also very interesting from the science point of view. So this is possible because we did this. If you ever try to do a mapping with mapping different environments and doing one environment at a time, you have struggled to try to figure out what is it the same? Is it different? Oh, but it's, you know, it's here, so maybe we can say it's the same thing. If you do this, you have a natural way of testing for this with all the environment combined. So global QTL can be detected as well as specific environment QTL by environment. And then I'm going to show you how we also use the same logic for G by E with genomic selection. And this is the work of another PhD student, Bettina Lado. And here we were interested in comparing strategies for exploiting this G by E when we're doing genomic predictions. And we were interested in, or we were wondering whether we could predict across locations, years, mega environments, or whether we need to evaluate everyone everywhere, right? Which is a huge investment in money. And so that was one of the questions we were interested in evaluating. For this, we had about 1,500 advanced wheat lines that were evaluated in 35 environments, and those were combinations of location and years. And they were evaluated for an 80K SNPs. And these 35 environments were highly imbalanced, which is very typical from breeding programs. So this data came from the Uruguayan breeding program. And you can see G by E here, where we had this in mega environments and the correlation across those environments in this pie plot. But this is hugely imbalanced data, which is very common. So we were interested if we could predict by year, if we could predict by location, if we could use this higher level of prediction for mega environments. And the model that we use is a very simple model. We use a GBLAB, M, we call it M for the average. If we're using just, you know, the Y is my mean across all environments, and then I have my kinship relationship matrix here and the genetic variance. But we also use a GBLAB G by E model, in which now my Y has all of this yield, let's say, in all the different environments. And now we are modeling this G matrix here as the Kronecker product of this relationship matrix times the correlation across environments, so like an unstructured model. And so with this, we were able to get predictions, and we did a few things. We evaluated for common number of genotypes because this was imbalanced. So we decided to do comparisons for a small number of genotypes, and that was 100. And here I have the predictive ability of the GBLAB, and the predictive, this is GBLAB M, and this is the predictive ability of the GBLAB G by E. If something is above the diagonal, that means that the G by E model beat it. If it's below, then there is no need to model it. And this was all for yield. And you can see that if we do it by year, so that means modeling the G by E across locations for a single year, the results are mixed. If we do it by location and modeling the correlation across years, this was better because most of the G by E was year to year variability. If we do it for the mega environment, you know, results were not very conclusive. They were very close to that 45-degree line. But what about when we go to larger population sizes The thing here is that we cannot do all of those year, location, and mega environments because we do not have the 900 individuals in each one of those. But when you compare it for the mega environments, first, you can see that the prediction accuracy, or predictive ability in this case, is way larger here than in any of these models. And that is mainly because we have increased our population size. So we have been able to increase the predictive ability by including larger population size, even when they are highly imbalanced across years and locations. So that's one thing. The other thing is that when we did these predictions and we were trying to predict by year, by location, we were looking at this mega environments. And what you can see is that if we predict for the mean of the mega environment, that means that if we are able theoretically to group locations in mega environments and then predict for those locations, what will happen is that, yes, we get a decent prediction accuracy. Of course, we get better if we have the actual environment screen. But for predicting new environments, this worked very good. So modeling G by E improves prediction accuracy, especially because larger population sizes can be deployed. But also, we can predict new environments if we rely on this mega environments or groups of environments that tend to have lower G by E. Then we can predict truly new environments. So that was one of the interesting things. And then what we did is we work a little bit on the same concepts in rice. And for this, we were looking at a population that had 637 advanced rice lines in 3 environments. And this was done with a 90K chips. And you can see some of the phenotypic performance for the different traits here. But I'm going to show the work of Eliana Monteverde. You probably know her. She is around. She's a graduate student of Susan McCutch, and we were working with them on this project. And what we were trying to do is 2 things. One is, okay, what if we were modeling this G by E? This was a 3-year data set. It was not as huge as the other one, but we were interested in looking at the performance of the different models. One with modeling the correlation across environments with an unstructured model, the other one with a diagonal model. And you could see that we did get good prediction accuracy when we were working on those unstructured models. Then we also included environmental covariates and use of PLS models. And you can see that we got a huge improvement in the predictive ability by doing that. So we can better inform our model. If we use the yield data to decide how my G by E structure is, it worked good. But also, if we use the environmental covariates, then the model was even better. So we can use those environmental covariates to get out of the, predicting new environments. We'll need to get beyond a yield measurement for that environment. So now, putting all these pieces together, right? We heard that a few things. Larger population sizes are better, right? Because we have higher predictive ability if we have larger population sizes. Then modeling G by E is very important. Replication and a good experimental design is also very important. So how do we put all of this together to optimize how we allocate the resources in the breeding program? And my thought was, this is what my breeding program looked when I got there to Wisconsin, was this. We had 6 testing locations. In each one of the testing locations, we were evaluating about 20 different genotypes in 3 replications. This is more or less what was going on. It was between 20 and 30, and it was 3 to 4 replications. But this means that we were using all of this 120 plots across 6 locations to evaluate 20 genotypes, which seemed to me like a huge investment and resources just to get a measurement on those only 20 genotypes. So I started to think about how do we improve this? And so what we work on, this graduate student, Pablo Gonzalez, and then another collaborator, Luis Diaz, we're working on how do we improve this resource allocation within and among locations so that we can actually with the same resources, evaluate more lines or have, a better response to selection. So the next step was, okay, people are doing this on rep. Remember, the correlation was not great, but we're evaluating more individuals. So my thought was, okay, is that balance worthwhile if we do this augmented designs, but balance across location, which is what most of the people are doing. If we have this set of 54 genotypes, some of those are replicated. I put them 6 genotypes replicated twice in each one of the locations and common across locations. So we will be able to evaluate G by E also and replication within. And then we evaluate the same genotypes in all the different locations, which is pretty standard. We said, okay, maybe we can do better by doing that. So we were interested in that question. But we also went a little bit crazy. And we said, okay, because we're confident that we can model the G by E. We're confident that we can model the spatial variability. So how about we know these mega environments and group of environments that have low G by E. So we grouped, based on historical information from my program, about 20 years of data, we created this consistent mega environments across the different years. And then we grouped them. This is mega environment 1, this is mega environment 2. And then we decided to create this kind of augmented design, but for the mega environment. So my thought was, if I have very low G by E here, I can use these different locations and I don't need to evaluate the same genotypes in all of those locations. I did replicate across my mega environments, but not within. And then I did have this checks, like an augmented or P-rep design. I did have my lines that were replicated in all of those. And then the final, we had some thought and said, maybe we need to be not so extreme. So we decided to have this moderate med. And it's the same general concept, but we put the blue ones here are replicated within an environment. So more replication within an environment, but not across environments. And that's a different shade of blue. So here is the code for what we have there. And we went on simulating all of these experimental designs, again, with uniformity trials. We were simulating, we know the spatial variability from the field based on actual uniformity trials. We know the G by E because we use the historical information to actually guide us in what the G by E structure was for that data set. So with all of that, we evaluated this with thousands of iterations throughout the field, moving, and for different sizes. And what we found is that, again, here I put the predictive ability. So this is the correlation between the true genotypic value that we gave the model and the predicted from the model. So this is predictive ability. And then here I have my 6 locations. And I'm showing you here the predictive ability for my RCBD, which is great, right? And this is a predictive ability in a different sense, in the sense that we're actually is the predicted value for that genotype, right? We have a phenotype, we have a genotype. We're not trying to do a CV cross-validation or anything. And you can see that, yes, of course, we're very good at evaluating. And that's why all the trials have been done with good replications. We're very good at saying what the genotypic value is if we put 3 replications here. So very good there. In this one, not as good. And this is the same as the first paper I show you, right? So not so good. This is not great. And you also have larger variance depending on the iteration that you happen to have. In the extreme med, we overdid it. We went very, very bad. So we didn't get any good predictive ability there. But in the moderate, you can see that, wow, this is looking good. And I'm only showing for the mega environment, right? Because we're not doing by environment prediction here. It doesn't make much sense there. So this was very good. The moderate med outperformed the balance on rep, this one, but not this one. So that makes sense because we have 3 replications here. So how about when we look at the response to selection? So now, because on this extreme med, I didn't emphasize that, but we have now 102 genotypes that we're evaluating here, right? So not 20, but 102. So my intensity of selection can get way higher for selecting the same number of individuals. So if we do that, and we have the predictive ability there, we have the variance, you can see that with my moderate med, we can have an increase of about 40% in my response of selection, just by allocating my resources differently. Okay, I'm using the same number of plots, but I'm putting them differently, and I can have a 40% increase in the response to selection. So this was very great news for us. And this is, we're now trying to figure out what the best way is to allocate this. And we went, very arbitrary in how we chose this initial study, but we're optimizing this. How do we actually optimize for how many do we have to replicate within? How many do we have to replicate across environments, but within a mega environment? So we're optimizing this strategy based on G by E and spatial variability for each field. So now I'm going to change gears, and I'm going to go a little bit faster on a few other things that are relevant for prediction. But one is, how do we predict the best parent combinations? And this is not predicting hybrid performance. So all of those work in hybrids. We're not predicting hybrid performance, but we're trying to see which ones are the parents that produce best progeny. And the mean of that progeny is easily, we can just get the mean of the two parents, and then we will predict the mean of the progeny. But what I'm mostly interested in is, can we predict this 10% superior progeny? And if the variance of that progeny is small, that 10% is going to be lower than in this case. So we did that in the wheat data set also. And the questions were, is it better to select the best parents or the best crosses? And do we need to model the fold variance if we select the best crosses? And we did it for several traits. And you can see here that for grain yield, there was no difference in how we selected those parents. And grain yield is a very complex trait. So eventually it averaged up, or you can say that the infinitesimal model works best, or very additive things they are happening. This is wheat, right? So we were not expecting dominance or other crazy things going on. But for grain protein, it paid off to actually model that variance and predict the best crosses instead of the best parents. And in breeding, breeders have always been choosing the combination of parents. They have never done this calling point, and I will cross everyone to everyone. They always chose who to cross to who. But then when we went to genomic prediction, everyone forgot about it, and we're just, crossing everyone to everyone else. So it pays off to do that, and we show it there. This is an extension of that, and we're showing, and this is not published yet, but we're showing how long-term response to selection is affected by these methods. And so very preliminary results, but we can improve the mean of the population by optimizing a few things here, and we need to control for inbreeding. But then we'll get very good response to selection. And then the last part is with replace phenotyping, which is, this concept of, okay, why do we want to do genomic predictions, or how do we actually implement it? Not just can we get a very good paper because we're doing this cross-validation one strategy. But one of the questions was with this wheat data set that we have multiple traits that we're interested in predicting, but especially the quality traits are very expensive and time consuming because, for knowing whether it's a good bread making variety, you need to actually bake it. So you create a dough, you raise it, and all of these crazy things that take a lot of time. So the question was, can we use this genomic prediction to allocate resources better in the breeding program? And what we did is we were here first looking at predictions for a single trait, multiple trait, 2, 3, or 4 traits. And you can see that, yes, there is an improvement in the predictive ability. And this is for the population size, just playing with both things. But there is an improvement in the prediction ability, mainly from 1 to 2 traits, but not so much from the other ones. These are correlated traits, right? So an improvement. But the most interesting result is this, that if we do a strategic phenotyping, so if we only phenotype a certain number of individuals, but not the same individuals for the same phenotype, so this is randomizing who do we score for sedimentation and who do we score for protein content, okay? and we optimize that. So we end up evaluating the same number of individuals for my training population, but in a strategically unbalanced design. Then we can see that with this strategy, we increase the predictive ability, and we can actually go down in population sizes to about 30%. So by being strategic in who to phenotype, we can improve the prediction accuracy by a lot. So these correlated traits, yes, animal scientists have done it for ages, but I think we can use them, and especially in this type of situations where some of those traits are very expensive to measure, and we don't want to measure all of them. We can be strategic in how we phenotype to increase the predictive ability. So with that, I would like to thank you all for listening to me, and I will be happy to take questions. Thank you.
Just for like general concept, what would like explain the difference between mega environments? This is that's maybe I'm familiar with.
Yeah, sorry, I didn't explain that. So the mega environments are defined as groups of environments with very low G by E within and high G by E across. In practice, most of the time, it is conducted based on the winning genotypes in each one of the environments. So if you have crossover there, then you call it different mega environments. If there is no crossover, the same winning genotype, then you define it.
So it's defined by the G by E selection, not by like environmental parameters?
No, it's defined by the G by E for that actual trait. So you do need the information from those environments, okay? In the case of our data set, where I told you the old data set, we evaluated the mega environments across the different years. What we did is for each year, for a series of 20 to 30 years, we said, okay, how do they group in 1996, the 6 locations? And I look at the mega environments. And then I went to the next year and said, how do they group? And then I look at consistency to see whether those mega environments were consistent across the different years. And in our case, they were most of the time. They were not perfect, but 8 out of 10 of the times, we get the same mega environments. So I'm pretty confident that those mega environments are true and not part of my imagination. And that we can, from next year, we will get a similar set of rankings within and among.
Thank you.
Yeah.
So you're defining mega environments based on or the actual performance, but if you go back and then look at the environments and what are the features and do they shift to some extent based on the sort of the weather features?
Yeah, so there are a few, and I show you a few different data sets here. So for the oats, it mainly has to do with the rainfall and whether it was a drought year or not. And in that case, I do have a little bit of shift in the mega environments. That's in the oats. In the wheat data set, it was not by location, and this was, Uruguay is a small country, but most of it was year to year. And what grouped them as mega environments was whether, so we first look at all the rain and all the environmental covariates to try to identify that, and it seemed that, yes, rain during grain filling or, at flowering was one of the things, but also all of the environments in one of the mega environments were non-fusarium head blight, no scab years, and the other ones were scab years. So I was actually able to predict for non-fusarium head blight years and for scab years. So in that case, it was very interesting. It's not easy to predict those because they are year-to-year variability, and we don't know if it's going to be a scab or non-scab year. In that case, in Uruguay, you get it 1 out of 7 years fusarium head blight. It's not like in other regions here where you get it every year. In the case of barley, it was mainly temperature. So that had to do with the temperature. There was some of the northern locations, northern is 200 miles north. So northern locations were hotter. And that created the possibility for a heat stress during grain filling. So how did that affect the leaf rust? Well, probably you get different races because of those different temperature and they are working on the races to see what are the prevalent races in each one of those. These were inoculated, but with a multi-race inoculum.
See if we have any questions online. Any questions from Geneva? Online? Oh. Pause for 2 seconds. Okay.
John or Ed?
Yes, so I was, people like me think you're learning speed breeding, right? And obviously there's some self things down the road with it. But if we actually started applying fast GS, how would you kind of integrate speed breeding, pushing 5 generations a year, or real crossing per year, where our ability to update our training models is going to be highly limited?
Yeah, so a few things there. Yes, if we go to that 5 generations, and we have just adjusted it for our oat and our greenhouse and all of that, if we go to that, we are going to be losing some of those LD relationship. We're going to be further away. So but we will not be able to update it until we put it in the field again the next season. So one thing is seeing whether we still keep how good is our production ability in those times. It will be compared to 0, right? So any gain there will be a gain because it will be compared to, we cannot evaluate yield in the greenhouse. So so then it ends up being a matter of cost and how much does that investment pay off on getting those. I think it's a good idea to try it. What they are also working on is, so most of the time, most of the breeding firms have implemented it in the sense that they are genotyping the fixed lines, right? And waiting until there. Those genotypes have a high value. We can put them in multiple environments. We can evaluate it thoroughly. But then we can also go on an animal model type of thing where we're having this cycling or breeding population that is independent of our variety development, where we're just taking from here, inbreeding, evaluating, and then releasing. So I think that it will be best served in that system, not just increase. So you could use it to just increase, and get to the F5 or F6 and get the F6 in the field. But I think that the most advantage would be in this recurrent selection type of scheme, where you do that. There are tricky parts there because one thing is to put those genotyping resources into lines that you have invested, you can replicate, and all of those. And I think that's the main bias of inbred breeders is that they do like to be able to put them in multiple locations. Whereas here you're investing that in a genotype that is going to be gone in 2 months, right? So it's a little bit more risky in the resources that you can do. I haven't done any work on how this model, I have been playing in my mind on whether it would make sense to implement it that way, or whether I'm going to transition into, inbreds and then go back there. It's and then
like your med model, you got 105 genotypes out there. You can update fairly quickly.
And that's actually small. We have more than those 60 plots, but yes, I think that's I think that genomic prediction, we're not going to get rid of the phenotype. We're just going to have to be better at evaluating the more promising, but we're not going to get rid of any phenotyping. We're going to do it better. We're going to do it more strategically, but we'll still have to put them there in the field to evaluate.
In one slide, you showed that the alpha design was better than the randomized complete block design, but I missed why, because I would assume that an incomplete block design would be worse than a complete block design.
So you said, let me rephrase, I'm not sure I heard you. So you said that the alpha was better, I said that the alpha was better than the RCBD, randomized complete block design, right?
It's surprising to me because I would expect an incomplete block design to be less accurate. Is it because the plots are smaller?
Okay, okay, perfect. I think I got it. But in the alpha design, we're working with resolvable incomplete block designs. So that means that I still have the 3 replications, and they can be grouped in these larger blocks, okay? So if I have 50 genotypes, right? In the RCBD, I have 50 in this block, 50 here, and 50 there, right? In the alpha design, I have 50 in this block, but within that block, I have incomplete blocks, okay, within this complete replication. But then I have another complete replication with incomplete blocks. The way it is optimized is that if a pair of genotypes are within an incomplete block in rep 1, I try, or the software does, to not put them in the same incomplete block in the next replication, okay? And then you do the same for the third one. So they are still, you still have the 3 replications, but now you have this within block control of the variability. And if you think about this 50 or 200, or any of the mapping populations you're working with, 400, 5,000, you cannot possibly put them in the same, in a uniform piece of land, all of those that will be one block, right? And the idea of the incomplete block is that it will help you to control for that spatial variability within. And the reason why I say it's better is because of the simulation results, not a priori. So we gain in everything. We gain on the best, on the recovery of the best genotype, we gain on the standard error, and we gain on the correlation between predicted and true values.
We have time for even 1 or 2 more questions.
Yep.
Yeah, thank you for the nice review. My question is, can you clarify a little bit the G block of the M, the simple mean of adjustment of the G value in G block?
Yeah.
Instead of just simple mean.
Yeah. So yeah. Let me go here and I will repeat. Okay, so the question was, what was the difference between this GBLAB and the GBLAB G by E, or the GBLAB M and the GBLAB G by E, right?
Also, M, is that just simple mean or just mean?
Okay. So this GBLAB M, what we do is my Y is the vector of mean performance. So what we do is, if we have a G by E study, right, where we have multiple environments and then replications or not, we don't care. What we do is we get the best mean estimate for the single genotype, okay? So I get one mean value for each one of the genotypes. So now the size of this vector is n, which is the number of genotypes by 1, right? So, so it's just that. It's a mean value based on a model that accounts for this, yes, experimental design, and accounts for the multiple locations. But it's just one single model, one single value for all them across all the environments. So mean across all the environments, okay? But it's a mean, it's not an average. So it comes from that model. And then the GBLAB, and this is assuming, like this is what everyone had done, right? When they get data, most of the people will just get a mean value for each genotype and predict it, right? They will summarize all the information from that study that they did in multiple environments as a one mean value for each genotype. And this GBLAB G by E, instead of having that, it will have a mean value for each one of the environments. So I'm always, and that's a very strong bias I have, I always use the mean values and not the raw data. And that is because I truly believe that the best representation that I can do of a genotype is the mean value in that environment that I can correct for my experimental design and all the spatial variability that I have. And that's my best bet at what the performance of that genotype in that environment is. So in this case, I have this, let's say now I had 50 genotypes evaluated in 10 environments. Here, it will have 50 means. Here, I will have 500 means, because now I'll have one mean for each one of those environments. Does that make sense?
This has been a production of Cornell University, on the web at cornell.edu.